Hello everyone. My name is Despotovic and I am extremely honored to have an opportunity to present our pilot study titled Genetic Analysis of SMAD4 C-terminal domain in patients with microsatellite stable early age onset colorectal cancer. I am a PhD student coming from the Institute of Molecular Genetics and Genetic Engineering, University of Belgrade, located in Belgrade, Serbia. SMAD4 is central mediator of the TGF-beta family of signal transduction proteins involved in many biological processes, including cell growth, differentiation, apoptosis, and migration. SMAD4 acts as a tumor suppressor and is commonly inactivated due to somatic variants in human cancers, most commonly in pancreatic and colorectal cancer. Loss of SMAD4 protein is a relatively common feature of sporadic colorectal cancer, and it was observed to be even more frequent in tumors of patients with early age onset disease and also more frequent in microsatellite stable tumors. SMAD4 pathogenic variant can cause loss of SMAD4 functionality or complete protein loss. Pathogenic variants in the SMAD4 gene are more frequent in the C-terminal domain and, in general, their functional consequences are more severe in comparison to the N-terminal domain variants. So the aim of this study was to perform genetic analysis of SMAD4 C-terminal domain in colorectal cancer patients with early age onset disease and microsatellite stable tumors. The study was conducted in a selected set of 20 DNA samples. Criteria for selection were no microsatellite instability and patient age at diagnosis of less than 50 years. Microsatellite instability analysis included 5 listed loci. Genetic analysis of SMAD4 C-terminal domain has encompassed analysis of exon 9 to 12 by PCR and direct DNA sequencing. The demographic and clinical characteristics of patients included in this study are given in table on the right. The tumor of one patient was found to harbor a SMAD4 variant in exon 9, and on the left you can see the genomic location of the variant, and below is the coding region. The observed C to T mutation leads to the amino acid change arginine to cysteine in the codon 361. This variant was discovered in a 47-year-old female with T3 stage carcinoma of the right colon, characterized by a low mucus production. The SMAD4 misses variant detected in our study was found in a patient that was the only one presenting with the right-sided colon cancer, which tend to produce symptoms only when they're relatively advanced, so it is probable that the disease onset of this patient happened way before she was diagnosed at age 47. According to the publicly available databases, this nucleotide is affected by alteration with a frequency of 0.2, 0.3, so it is relatively common in the general population. This variant has been functionally characterized before as pathogenic in few studies. The detected variant is located in a hotspot of missense mutations, which disrupts binding of SMAD4 to the other SMAD proteins. Actually, the crystal structure revealed that the detected SMAD4 variant maps to the loop helix domain of the C-terminal domain of SMAD4. The functional effect of this variant is prevention of hetero and homo oligomerization of SMAD4 and suggests that the mutated SMAD4 protein, which is not bound into couples with other proteins, is degraded or unstable in vivo. So, to conclude, we started from the fact that the pathogenic variants in the SMAD4 gene are more frequent in the C-terminal domain, and we did observe a C to T variant in SMAD4 exon 9, known as a mutational hotspot in one individual with early age onset colorectal cancer. The variant is responsible to the amino acid change arginine to cysteine, known to disrupt SMAD4 oligomerization. Considering the incidence and functional consequences of SMAD4 exon 9 variants, the screening of this region could be a useful low-cost strategy for genetic analysis of colorectal tumors from patients with early age onset, as well as for susceptibility testing. Absence of microsatellite instability in the tumors and their presentation as right-sided lesions could be additional factors to be considered as parameters for selection of patients for this genetic testing approach. Thank you very much for your attention.